Welcome back, class. Welcome back to the lecture, Genomic Marker, lecture 16. We'll be talking about one of the technique, polymorphic marker technique called RAPD. It's pronounced as RAPID. It's abbreviated as Random Amplified Polymorphic DNA. So we will look more details into the molecular mechanism of this polymorphism in terms of how they will form and what technique we have used in terms of molecular biology, whether we are using restriction digestion here or we are using blotting technique here, or we are using simply just a PCR with primers. We will look into that. And as discussed in the last class, end of the last class, the comparison of three important molecular polymorphic marker or molecular marker helping to identify polymorphism. We looked in two markers, REF-flip and A-flip. These two are common, having the FLAP. Alphabets are common, only R is differing and A is different here. Basically, that explains, the name itself explains the technique or methodology you are using. You are cutting it here, the given genome on restriction fragment sites or restriction enzyme sites based on the restriction enzymes and recognition and binding pattern and cutting pattern. You are breaking them, and after breaking, you are running through them agarose gel, and specifically to look into your REF-flips or polymorphism in a given location you are looking, you design specific DNA probes, you undergo a plotting technique and hybridization Southern blot to visualize the important features of your own. On the other hand, A-flip is nothing but R is replaced by A. A means wherever A is there in this molecular marker technology, we are looking for amplification. Once you say amplification, you are basically using a PCR, polymerase chain reaction. But A-flip is a combination of REF-flip plus PCR. So you already have a restriction enzyme site, you digest them with that. After that, by knowing taking the advantage of restriction site or sequences by knowing yourself, you can design a primer according to this vicinity of this restriction enzyme sequences. Either you go up or you go front. You need to recognize the sequence. You make specific adapter molecule on the back side. On the front side, you restore the restriction site if it is broken as sticky end. Then you continue with pre-amplification and then further amplification steps, adding specific nucleotides for the primers. And the primers from this side and that side will help you to sequence the genomic sequence region in between that. So you can analyze that difference if it's there in terms of the size. So that's a REF-flip and A-flip. Coming on to this technology, though it has R and it has A, it doesn't mean that you are mixing restriction digestion and amplification again, PCR. It is R in terms of rapid is another English word that's called random. So it says random amplified polymorphic DNA. So it doesn't use restriction enzymes. Please don't get confused with the R. Wherever R is there, it's not restriction enzyme. In this case, it is amplification. It's a random amplification. So it uses only PCR amplification, no restriction digestion. Once you amplify the PCR product, then you separate it by the gel electrophoresis. Simply visualize the electrophoresis or amplified product from PCR through binding with ethidium bromide. No Southern blot involved, no restriction digestion involved. A simple PCR step is involved. As suggested previously, these two are dominant marker. This is a codominant marker, saying that heterozygous situation or allelic variation you cannot identify. Given the homologous chromosome or given the two alleles, you cannot separate the alleles here because irrespective of the whatever change in between, this simply amplifies the DNA in between blindly. It doesn't care. It has how many restriction site, what is in there. Here it will cut the restriction site and you take site 1, site 2. Between that is genomic DNA. Here it doesn't take restriction enzymes. You give primers. It blindly amplifies the region in between that. So based on this, you cannot distinguish. It cannot distinguish the methodology inherently any changes in between that. So in A-flip, selective primer as suggested for PCR. You know the restriction site. You design a primer knowing this sequence, so you have the control. But in RAPD or rapid, you don't have control over the primer. You can pick some random sequences of ATGCs and make a primer out of it and see where it binds randomly in the genome. We will look into that. Small examples we will look. And here, A-flip, it involves only both restriction digestion and both PCR. So each step involves a starting material, that is purified DNA, and a restriction digestion usually requires a DNA in micrograms, and then PCR requires in nanograms. So you need a large quantity of purified DNA, high quality of the DNA should be there to start this technique. And the results are highly reproducible here because you have control over the sequence. But coming on to rapid, you may, because you're going with PCR alone, nanograms quantity of DNA is enough, and you may have low reproducibility per se. We will see that why. So just first stick to that. It's a technique based on random amplification of polymorphic DNA using PCR. So basically saying that you have to design some primers. The primers are random. We will see that what is the randomness. So the whatever I have spoken in the previous slide, it's again you look into the genomic diagrammatic version, different restriction enzymes, different location in the genome over 35 kilobase span, and the sites may be polymorphic given each restriction site, or this site may be polymorphic to human 1 to human 2. That's why it gives different fragments or size on the agarose shell. Okay, let's take our golden slide. Typical pattern, we looked always different concepts. We studied looking into this blank DNA sequences. In this slide, you're just looking ATGCs only, no more new alphabets, only ATGCs are very randomly arranged. So it's a fundamental to human DNA. It's a lot of junk, lot of repetitive elements are there. For example, TTT I'm seeing here, TTT, 3 T's I'm seeing here, 5 T's here, 3 T's again. So you may have a repetitive element of a given nucleotide there. That is really important for one of this rapid to identify polymorphism. So we will quickly brush up on fundamentals again. We will talk each step by step slowly. DNA amplification. For example, these are the steps we will discuss in the next few slides. DNA amplification, primers, PCR, amplicons, agarose gel, and sequencing. These two we already covered. It's no need to talk. But take an examples upon these concepts. So let's say, for example, I want to amplify the given DNA here. So I don't know what the length of the DNA here, but I know the sequence given the ATGCs here from starting C and ending with G at the end of the slide here. I know all the sequence information here. So I want to use the PCR technique to amplify this DNA sequence on the background here in the slide. So the first step, what I will do, I know the sequence, I know the start, I know the end. So I have to design the primers. Oh, I will design a primer here called forward primer. I will design a primer here, it's called reverse primer. Remember that DNA amplification is assisted by DNA polymerase, and the polymerase can add a nucleotide at the end of the 3' only. So 5' 3', this is 5' 3', reverse primer. So basically you move in that direction, you come from front, you come from back, you design a specific forward and reverse primer. Based on that, you amplify the DNA given here using PCR reaction. That is the typical way you go in terms of that. So let's say, what is the length of the DNA here in terms of the nucleotides? How many nucleotides are there? Usually it will be a double stranded. We will show only one strand here for simplicity, but it's all double stranded DNA you are looking. Each line column here will be a complementary DNA should be there on the other side. So considering this length here, how much is the length amplified by this primer set, forward and reverse primer? What is this, 10 kb, 20 kb, or 3 kb, 5 kb? If we approximately look here, the first column, the column length is, I think, approximately 150 base pairs or 150 nucleotides are here. So if I count the columns from top to bottom, I am seeing almost 150 columns here. So 150 nucleotides in each column and 150 columns, the typical length. Sorry, it may be, yeah, we can change here. It may be more than 6,500 base pairs. For example, so we are saying 150 into 50. Okay, sorry. So how many columns are here? It may be 50 columns here, not 150 columns. It's okay, we are right. Second, so you are looking in a column of the 50 columns. Each column will be having a length of 150 nucleotides. So total length of the DNA given here is approximately 6500 base pairs. Okay, that is the typical we are looking here in this background slide. I have a forward and I have a reverse primer. We'll see that whether if put in the reaction in the PCR, whether this reaction will proceed, we will get the 6500 base pairs or not. So I'm simply saying that the region between two primers, forward and reverse, amplified by PCR is called amplicon. And amplicon is the region forward amplified, DNA amplified between forward and reverse primer, given that you know the specificity, you know the location of the genome. So usually in PCR conditions, PCR normal conditions, typical length the primers forward and reverse can amplify is limited. Usually it's 1000 base pairs only. And it's a PCR, it has a technology you are using in vitro, you are adding all the components, we will see in the next slide. So the typical length is 1000 base pairs only. So ideal amplicon length depends on many variables and design preferences of a PCR technique. For standard PCR, scientists usually do a PCR where they can amplify only 200 or they can amplify maximum 1000 base pairs. That's a typical they use. And for quantitative PCR, qPCR, it is more shorter, 75 to 150 to know about more changes within the given sequence. So it's a qPCR. In normal PCR, maximum 1000 base pairs only. Why is that? Because a typical PCR reaction involves a different components because you are recapitulating DNA replication process from in vivo to in vitro. So you are using in a test tube, you are amplifying the given DNA on the background here. So you use a specific components, buffer, DNTPs, nucleotides, magnesium for enzymatic reaction, primer 1, primer 2, and also the primer concentration, Taq polymerase, and the template C 10 nanograms per microliter only. And then you water, you dilute it into a specific PCR reaction, 25 microliters. Then the final concentration also is limited. You may use only 1 unit per reaction of Taq. You may use only 0.25 micrometer of primer so that the volume, the components are less so that there is more specificity. And DNTPs, given the nucleotides also, you can use 0.5 millimolar only. So saying that 0.5 millimolar may give some nucleotides, maybe 1000 or 2000 nucleotides only, or maybe 500 nucleotides only, DNTPs, so that only 500 base pairs of reaction can proceed. Inherent to the PCR technique, the amplicon length is always typical 1000. If we take the scenario, we have the background DNA here, the length is 6500 base pairs, then can it be extended by PCR given this forward and reverse primer? Both are good, both are attaching complementary to the given DNA. If you run a PCR, basically the reaction will not proceed. It will not proceed, though primers are there. It will not give an amplicon, it will not show any PCR product on the gel because the primers red and blue cannot extend 6.5 kilobases because there are not enough nucleotides. There is not enough polymerase present in the reaction in vitro, so the reaction will not proceed. Let's say you move this primer to somewhere here, the blue one, then the typical length is 1000 base pairs, then the reaction will proceed. Surely it will proceed because you have provided the enough components for the reaction to proceed with that. So remember that fundamental difference in the technicality and components of PCR. If given the primers are good, complementarity is good, the reaction may not proceed. That is really fundamental for RAPID also. We will talk. So now we are looking in a length of, what is the length of amplicon produced by these two primers. Now I am asking another question. What is the typical length of the primer you should use? What is that? You can use 50 or 100 base pairs, 100 nucleotide length primer. So why I am cutting the primer here? I can extend the blue up to this till point this here, end of the column. I can use red from here to end of the column here. Can I do that? No, absolutely you cannot do that because the reaction has some typical primer binding, typical annealing, and temperature is common to the given composition, JTGCs of the primer. Given the nucleotide, you cannot use such bigger length of primers. you use typically length of the primer is 18 to 22 base pairs only. That's why it almost 19 here. The typical length is 18 to 22 base pairs, and it is generally accepted that optimal length is this one. This length is long enough to for adequate specificity. For example, I am trying to amplify this region here only, nothing else. So my forward primer should have maximum complementarity to the given sequence here. Otherwise, it will go randomly bind somewhere in the genome. So I may not get the gene X here I am looking for. Same way, reverse primer also should have maximum complementarity here. So the specificity of the primers, given the location of the genome, what gene you want to amplify through PCR, the primers are really important. There should be highly specific primer, and also length should be maximum, and also typical these five nucleotides at the end of the primer here should have maximum GC components, GC nucleotide over 80. By doing so, first this 5 nucleotide complementarity primer can bind, then the remaining nucleotides come back in the back as a zip, and then they keep stitching it up or complementarily binding. So these typical 5 are important, so per se. So length is important of the primer also in terms of specificity and complementarity. They are good, highly specific. If not, non-specific amplification or random amplification happens that you don't need to, that you don't want, given that you are really looking for a chromosome X. Location is this because I want to study a disease condition in the wild type and disease scenario. I want to know the wild type amplicon of this. So I want to do the experiment in a normal, proper condition. So let's say now I have two primer sets here, red and blue. One is longer, typical length. Another one is shorter. I made it short. So of these two, which will be highly specific? No doubt, the longer will be highly specific for a given gene or locus here. Given that, it will go and bind it. So why I'm not saying this small, let's say 12 nucleotides here, why I'm saying it is not, probably may not be specific? Because if I look into this band here, I look into this complementarity here. So let's say it is AGTG, ACTG of this sequence. This is another sequence here. Within these 6,500 base pairs, I might duplicate this. I might have a same sequence, exact copy in another location here. This is another location here because I'm shortening the DNA's nucleotide length. It may have more binding because more repetitive elements are there. Or if I make it more shorter again, small, I may get more frequent binding of these primers, not only here and here, maybe in different locations and randomly in a given 6.5 kb. How is it happen? Just like this. So now you have different lengths. One is highly specific, complementarity, forward, reverse, typical PCR. That is good enough to go for PCR if given the typical length. And the medium and short fragments, because given the repetitive nature of the DNA sequences here, or given the human genome, lot of junk and repetitive elements in the non-coding, a small fragment of a primer, if you are making a small primer of any sequence, not specific, any random sequence of ATGCs you take here, you make a small primer of 8 to 12 nucleotides, it may go and bind randomly in a given genome. And this randomness is fundamental to understand the polymorphism also. We will see that. Now, in this slide, I am showing two colors, forward and reverse, red and blue. I'm showing two different primer sets, two different sequences, one forward, one reverse direction. And smaller the fragment, more randomly it will go and bind the DNA, given the DNA. So, let's say I make it all red or all blue. I'm making all red here now, leaving the typical PCR primers, long complementarity primers of 20 base pairs. These primers are less than 8 to 12 per se. So I'm making only one color now. I'm not making red and blue. So I made a primer with the same sequence randomly ATGCs, and I used the same primer as forward and reverse when I'm doing that reaction. The directionality of the primer changes if you make a primer. So if it's 5' to 3', it will go and bind this. But if you make a sense and anti-sense strand, it will go and bind the sense. It will go and bind the anti-sense. This direction I'm talking is I'm looking in the double stranded DNA here. You duplicate each column. One primer goes on the top, another primer goes on the bottom because it's a bidirectional replication process. So given the same primer I use, random sequences of ATGCs, given a genome here, it may go and bind randomly in the given location. Then if I run a PCR, for example here, what do you expect? What do you expect if you run a PCR using only one primer in both forward and reverse direction? What will happen in the PCR reaction? It will happen. And let's take an example here. So this is one genome, or let's say chromosome 1 or chromosome X. The same primer randomly I picked ATGCs. I have chromosome 1, 2, 3 and 4 here. I used the same primer and I allowed the primer to randomly anneal in the PCR reaction, go into different portions of the chromosome 1, 2, 3 and 4. So if I do that reaction of annealing, given the specificity, given the randomness of the genome, the primers both forward and reverse can go bind randomly in the different location of the genome or different chromosomes. Typical chromosome 1, they may bind here in forward and reverse direction. There are more forwards here than 2 reverse, 4 forward. It may be a typical pattern here, we'll see that. In chromosome 2, no binding at all. Or binding is there in terms of annealing, the primers forward and reverse are too far away. Only one primer site was there, they are too far away. Let's say this annealing situation. Chromosome 3, you have 1, 2, 3 patterns. Chromosome 4, you have 2 patterns of annealing, forward and reverse primer. Remember that the primer is only one primer set, only one primer. Both forward and reverse primers are same here in a given experiment here. So they annealed here randomly. And now I am amplifying in terms of the PCR. I have given enough nucleotides, enough polymerase. What will happen to it? How many bands I can expect in a PCR reaction? This situation, chromosome 2, they are too far, maybe 6.5 kilobases apart, based on the PCR inherent problem. Though there is complementarity of the primers, the band cannot be produced given the PCR reaction. So no band here in chromosome 2. On chromosome 1, I'm seeing that forward and reverse, more forward, less reverse. But I'm looking for co-localization of a forward and reverse primer given the same red primers. So typically in a given condition, I may amplify this set only. I may not amplify this and there is no reverse primer in vicinity. Maybe this is the most suitable amplification that goes in PCR. Blindly it picks and depending on the location, it amplifies nucleotides. You may get one band only here. And in chromosome 3, very nice pattern. They got and bind different locations, very close to each other, forward and reverse. So you may get 3 bands here. Even chromosome 4, 2 bands here you may get. So this is nothing but typically you are speaking a rapid technology. In rapid technology, how do you distinguish 4 chromosomes of a given human being? You design a specific random primers. You use the random primer as forward and reverse, both same, and then you start annealing it in PCR reaction to the all genomic sequences based on the location of this primer complementarity. As the primer is 8 to 10 nucleotide only, it will find a lot of complementarity sequence given the repetitive nature of the genomic DNA. But that repetitive nature also can vary from chromosome to chromosome based on the polymorphism or variation between the chromosome. The primers bind randomly and they give different bands in the agar-gel pattern after PCR. So this is that rapid technology, a fundamental mechanism. You are talking about a chromosome in a given human chromosome, a given human 4 chromosome you are talking. But if you take 4 different human beings, for example, in biotech department, myself, Girish, Priyanka and Santosh, these are the 3 genomes of ours. And then one more example is Prasanna Kumar, chemistry HOD. So we ran the same primer, we selected the 10 decamer primer. We ran the PCR annealing. Depending on the variation among human beings or polymorphism among human beings, I'm different from Priyanka, Priyanka is different from Santosh, we both are each other different. And given that there is a primer binding specifically, and the gel will show different bands based on whoever is it. Based on 3 bands, it is Priyanka, Based on 2 bands, it is Santosh. Based on 1 band, it is me. And based on no band, per se, we are all biotech. He is a out of the guy in chemistry department. And luckily, the gel also not giving any band at all. So 1, 2, 3, 1, 2, 3, we can identify biotech people. And no band at all. Prasanna out of the chemistry people, no band, no poly. So basically, the polymorphism within a department, different departments, based on these primer binding randomly, you can identify. So that is a typical technique when you speak of rapid. The rapid RAPD is pronounced as rapids or DNA fragments amplified by PCR using short synthetic primers, generally 10 base pairs of random sequence. So you don't know, you basically no need to pick the ATGCs typically looking into the DNA genome, because in rapid, you don't need to have a starting genome material, no genome sequencing, you don't need. Randomly, you take ATGCs, make some 4As, Ts or Gs, randomly you can make a primer and see that whether it's a primer has efficiency in binding the different genomic fragments or not. So typical 10 base pair random sequence of the primer will allow you to analyze the different DNA fragments amplified by PCR. So these primers, oligonucleotide, they are the same for both forward and reverse, except the directionality you move. You make a primer 5' to 3' forward. In a reverse primer, you reverse it. You make that in opposite direction, so that you are synthesizing two different primers in terms of direction, but the composition is same. You given that primers, both forward and reverse same, they can go and bind some a common pattern of given information of 1 to 10 genomic sites simultaneously, because not more than that, given the shortness, given the complementarity, given the PCR reaction. The typical limitation of the rapid is that they may give one chromosome, in one chromosome they may give one gene only, or they may give 10 loci. So 1 to 10 genomic sites, they can go and bind simultaneously, and those products can be analyzed in the PCR. So the typical length is 0.5, means 500 base pairs to 5000 base pairs. That is the typical length they amplify using these random primers. And based on the amplification, based on the differences, for example, presence or absence of bands, particular sizes. Presence means 1 band, 2 band, 3 band. Absence means no band at all. That relates to the polymorphism of populations. You can identify that. So basically saying this, primers are important, random primers, and the annealing sites are annealing. Primer annealing step is also important. Initially people were thinking rapid is mainly based on primer annealing sites, the given genome. Even the repetitiveness, they may go and bind. But the more they look in the PCR, they see that not only the binding, the vicinity where forward and reverse primer are available within the shortest possible distance, that also really important. That's why the generated length differences in the amplified sequence also important. Not only annealing of the primer, the difference between forward and reverse primer location and how much of the sequence they can amplify, that is also really important criteria for rapid. So those points again explained here, different colors, you go and look into that. The method again described here, it's nothing but used for the random amplification. And what are the steps involved per se? You go into that. First, you need to isolate the DNA of the given species, and then you add different primers. That primers nothing but, you add different amount of the oligonucleotide primer, saying that the primer is 10-mer only, decamer, and you add large quantity, the excess quantity of the primer, so that they can bind in a given reaction enough genomic sequence. So then you continue with the same typical PCR steps. You will follow the endpoint, ethidium bromide binding in the agarose gel. You simply follow these steps, step 1 to the step n, starting from isolation, how the PCR reaction you do, annealing of primer, what temperatures you use, and then going to the end of the product. And these steps are important. If given a rapid as a 15-mark question, you need to write all these steps. So you go into that. So that's a basic protocol for that. And given another example here, typically whatever I showed in the blank nucleotide sequence in the slide, same way, a person want to use the rapid technology, you distinguish chromosome 1 to 7 of a given human being. So this is chromosome 1 larger, 2, 3, 4, 5, 6, 7, and he designed some random primers, decamers, and he started annealing the primers to these 7 chromosome. And not a typical situation. One situation of that in the chromosome 6, no binding site, primer annealing sites are there, no binding happened here, no product, so 0 band. So here, and but in 5 only one reverse primer was able to bind, no forward primer. Again, no PCR amplification happens in this region, no band will come. 5 and 6 bands are out of the picture, but given the other chromosome, the chromosome 1, they were able to see that randomly 2 sites it's bound, and one site is giving longer fragment, shorter fragment, shorter amplicon. Another site is giving a longer amplicon. Basically typical on the size and the agarose gel, smaller goes always into the bottom. So A is typically here, and the B is the longest or largest given the any amplification here, that is on the top, and DCER random amplification happening in the other chromosome. So that is the technique RAPID can use within human population of chromosomes to distinguish. So of the 7 chromosome, you may not get 7 bands because 5 and 6 are absent, but other bands are there, A, B, C, D, E, based on the size. So this is again in terms of the descriptive, you have to see into this descriptive nature, what is it important for. Look into that dominant and multi-locus. I explained already in the previous class. Look into that. Then typical mechanism, how it works, again explained here. One more important, that the same RAPID technology used to generate gels, and if he's a supervisor or principal investigator who has big experience in this PCR, his gel looks like that. But if the same procedure, same primers given to a starting undergraduate in the lab, he will do the same experiment, his gel looks like this. We cannot blame the guy. He is a beginner. He may not have done proper conditions in the given, too much of tubes he has to manage, too much of a microliter, 0.5 microliter of DNA polymerase. So 20, 30 tubes are there. Maybe he is not dexterous with his hand. So it may not happen. But typically the gel should look like this, because you are looking for a difference in the band pattern. So if the gel looks like this, how do you distinguish from mutant to wild type? So you need a typical length that score highly efficient gels. So in another case, you are looking for 2 polymorphism, human 1, human 2, DNA of human 1, DNA of human B, let's say template A, template B. The primers go in 2 patterns here, they may go in a pattern here. So only these 2 can amplify 2 different fragments. This can amplify only 1 fragment, not this only 1 primer in one direction. So gel looks, A has 2 bands, B has 1 band. This is typically a polymorphism. It may be human 1, human 2. It may be disease condition. This may be wild type condition. It may be a disease condition, mutation happen, some deletion of the sequence. That is the way RAPID helps to see the polymorph. So you look in terms of strengths, you look into that. There is a low quantity of DNA, PCR advantages. You look into the weaknesses, you look into that. One point is that so weakness is one of the biggest weaknesses, reproducibility. For example, maybe the PI gets this gel, being the expert in one experiment, he gets this kind of pattern. Then 1 month later or 2 weeks later, he just repeats the same experiment to find the gel again. Somehow the primer didn't bind properly, annealing temperature was somehow random. So basically he may not get the same gel again. So you need to repeat it several times to get a reproducible pattern. That is one of the problem weakness in RAPID. You go and look into that in limitations. This is one more technique called SCAR, where you don't need to look in the heterozygosity in the given RAPID, but you can amplify a specific DNA fragment here and clone the DNA sequence, identify the mutation. Then you can look in the heterozygous situation also. That is a SCAR technique. And in terms of application in the RAPID, EAP-PCR, DNA fingerprinting is another technique. Here long primers are used. You look into the normal condition, disease condition, how the gel differs. Look in your free time. This is nothing but DNA fingerprinting. Okay, you look in that, look in the diagram specifically what are the bands. We might take this diagram for next class on microsatellites, SSRs. Tomorrow we will finish SSR. Okay, thank you.